everybody. Welcome to Biology in the Kitchen. And today what I want to do is I want to talk to you about photosynthesis. And I have a really weird and fun demonstration to show you. So what I'm going to do is I'm going to take this geranium plant and we're going to do a photograph on a leaf. The ultimate photosynthesis. We're going to do a photograph on a leaf. And what I did is I took a picture of Abraham Lincoln, and this is called a negative, where all the white is black and the black is white. This is how they used to make photographs in the old days. And anyway, here's a young Abraham Lincoln, and you can see some areas are clear, if I show you that, and some areas are clear and some areas are dark. And what I'm going to do is I'm going to put this on the leaf like this. I'm going to fold the foil over. And I'm going to try to block out any of the light so that the only thing that can get through to the leaf is what can go through those clear areas in the photograph or in the negative, okay? And so then we're going to put it underneath the light and we're going to let it sit there for about 2 days. Usually I set this up on a Friday and then we finish the demonstration on a Monday, okay? So to make this all happen, so then we're going to do that. I already set this up a few days ago, and just to hedge my bets, I did a few more photographs as well as Abraham Lincoln. And so here's Abraham Lincoln, and this is Queen Victoria and Prince Albert, and here is the Men in Black. So let's do Abraham Lincoln because that's kind of what I had in mind. And we're going to pluck the leaf for Abraham Lincoln off of here. And we're going to unfold it. Now think about it for a second. What do you think you're going to see on this leaf? Do you think there's going to be like a green Abraham Lincoln, or do you think that that's just going to look like a normal leaf, or do you think there's going to be some kind of like a halo sort of area where you can just barely see Abe showing up? What do you think? All right, let's take a look. So we're going to pull it out of the foil, and voila, absolutely nothing. Does that mean the lab failed? Yeah, no, watch. All right, so now what we're going to do is we're going to take this leaf and I'm going to boil it in water. We're going to put it in water for 1 minute. Now that does two things. Number one, you learned about enzymes, and it kills the enzymes, or it denatures the enzymes so they can't work anymore. It stops all the chemical reactions, okay? The second thing that it does is it basically cooks the leaf. That softens all the cell walls so that the next two reactions that we do, those chemicals can soak into the cells and make that happen, okay? So we're just gonna cook the leaf for about 1 minute. And then what we're gonna do after that 1 minute, then we're gonna put it in another beaker over here that has alcohol in it, methanol to be exact, all right? And methanol is a solvent, okay? I think it's been about 1 minute. Okay, so I'm just gonna pick up this leaf, all wilty and cooked, all right? And now I'm gonna put it in the methanol. And we're gonna boil it in methanol for 1 minute. Now, methanol is a solvent, all right? And what it will do is chlorophyll, it turns out, is kind of like an oily protein. And so that methanol is a solvent for the chlorophyll, and it will cause the chlorophyll to leach out of the cell. With those soft membranes, the chlorophyll will just go bleh and just come right out of the cells, all right? So we're gonna boil it in the methanol for about 1 minute. And while we're waiting for that to happen, I want to review with you a little bit about photosynthesis, okay? Now this is stuff that you learned in like elementary or middle school, but we're going to take it up a notch, okay? So you remember all of this. Photosynthesis happens in the chloroplast, okay? And inside the cell there are hundreds and hundreds, I want a little bit more time, hundreds and hundreds of chloroplasts in every leaf. And each one of those little stacks inside there, called thylakoids, is where photosynthesis happens. So this is like a huge battery that's going on. And the sunshine hits the leaf and hits the chloroplasts, and that activates the enzymes. Oh, I forgot my pen. Enzymes. All right? And that'll activate enzymes inside the chloroplast as that sunshine warms it up. Okay? So that's the first thing we're going to talk about. All right, so now the leaf is ready. And now we're going to take the leaf and we're going to expose it to our good friend, IKI, iodine. All right, so here's some iodine. And look at that. Look at the leaf of how it lost, not all, but a lot of its chlorophyll is gone. And we're going to soak it in the IKI, and this takes a few minutes. And then we'll take a look and see if we got a photo of Abraham Lincoln. But let's finish this. All right? So, in photosynthesis, carbon dioxide comes in from the air. You guys know that. Water comes up from the roots. And the enzymes and the energy from the sun bust this up so that you've got these carbons and you've got oxygens and you've got hydrogens all roaming around inside this chloroplast. It's the enzymes that actually knit it together and make glucose. See, carbon in, carbon out. Hydrogen in, hydrogen out. Oxygen in, oxygen out. And yeah, there's some surplus oxygen, and that's why we always say plants give off oxygen. It's a waste product for them. Now this glucose goes on to make several different things. It can be knitted together to make starch. It can be knitted together to make lipids, like, you know, olive oil and canola oil and peanut oil. It can be made into proteins. And of course, it can be made into DNA, nucleic acids. All right? So we're going to test it to see about the starches. It's off. I turned it off. All right. Here we go. Let's see if Abraham Lincoln showed up. There he is. Oh my goodness, this is one of the best ones I've ever done. I did get a Harley Davidson on a leaf once. That was pretty cool. Look at that. So all of the places that the light could get through, they went through photosynthesis. They were able to convert carbon dioxide and water into sugar, and then they stored it as starch, which of course turns black with the IKI. And all the places that were light, that were clear colored, that's where the photosynthesis happened. But all the places that were black, they blocked the light, and therefore photosynthesis couldn't happen, and those places stayed, well, white, but you know, brown with the IKI. But anyway, there you go. Abraham Lincoln on a leaf. The ultimate in photosynthesis.